Welcome back, everyone. In the previous lecture, we built a strong understanding of biological oxidation and oxidoreductase enzymes. We learned how electrons are removed from metabolic substrates and how enzymes safely transfer them to prevent oxidative damage. But what happens to those electrons after removal from glucose, fatty acids, or amino acids? Do they just disappear? Absolutely not. Every electron carries potential energy, and cells capture that energy with an efficient system called the Electron Transport Chain. Also known as the Respiratory Chain, the ETC funnels electrons through protein complexes to ultimately reduce oxygen and drive ATP production. In this video, we'll visualize complexes 1 to 4, show proton pumping across the inner membrane, and watch ATP synthase make ATP step by step. First, a quick anatomy reminder. Mitochondria have an inner and outer membrane, an intermembrane space, and the matrix where many reactions occur. The ETC proteins are embedded in the inner mitochondrial membrane and arranged as complex 1, 2, 3, 4, plus mobile carriers ubiquinone and cytochrome C. Complex 1 accepts electrons from NADH. Complex 2 accepts electrons from FADH₂, and funnels them into the same chain via ubiquinone. Ubiquinone, or coenzyme Q, is a mobile lipid-soluble carrier that shuttles electrons between complexes within the membrane. Cytochrome C is a small water-soluble protein that carries electrons between complex 3 and complex 4 across the intermembrane space. Complex 4 reduces molecular oxygen to water using electrons donated through the chain, completing the electron transfer process. Electrons from NADH enter at complex 1. As they pass through, complex 1 pumps protons from the matrix into the intermembrane space. Electrons from FADH₂ join the chain at complex 2, which does not pump protons but passes electrons to ubiquinone. Ubiquinone carries electrons to complex 3. Complex 3 transfers electrons and contributes to proton pumping via the Q cycle mechanism. From complex 3, electrons move with cytochrome C to complex 4, where oxygen is the final electron acceptor and water is formed. Proton pumping totals: roughly 4 protons at complex 1, 4 at complex 3, and 2 at complex 4 per pair of electrons from NADH. Net effect: NADH oxidation drives about 10 protons out per NADH, creating an electrochemical gradient across the inner membrane. The proton gradient stores potential energy called the proton motive force, which ATP synthase harnesses to synthesize ATP from ADP and phosphate. Protons flow back into the matrix through ATP synthase, driving rotation of its subunits and mechanical generation of ATP molecules. Biophysical estimates give roughly 2.5 ATP per NADH and about 1.5 ATP per FADH₂ in typical mitochondria under physiological conditions. In short, electrons drive proton pumping, the gradient drives ATP synthase, and chemical energy from food becomes usable cellular ATP. We can visualize the whole process with step-by-step overlays. Electron path, proton pumping points, and ATP production highlighted for clarity. These visuals help students connect molecular motion to energetic outcomes, how microscopic transfers create macroscopic cellular energy supply. The ETC is tightly regulated by substrate availability, ADP levels, oxygen supply, and feedback from the cell's energy demands. Specific inhibitors block the chain. Rotenone inhibits complex 1, antimycin A targets complex 3, and cyanide blocks complex 4, halting respiration. Uncouplers collapse the proton gradient, allowing electron flow without ATP production and generating heat instead of useful chemical energy. Clinically and physiologically, proper ETC function is vital for energy-demanding tissues like heart, brain, and skeletal muscle. Quick recap. Electrons from NADH and FADH₂ travel through complexes 1 to 4. Protons are pumped to build a gradient, and ATP synthase converts that gradient into ATP. Thanks for watching. Review the labeled diagrams and step overlays, and pause to study each complex until the flow and energetics feel intuitive.